This is going to be a video on cellular respiration. This is what I like to call big bucks and big bonuses. A lot of college students that end up taking biology don't really necessarily understand the whole entire component and diagram of this. So this is where I like to say is going to be a key component when dealing with cellular metabolism, and this is going to be with the without the protein, starch, and what I like to consider other metabolisms out there. So what I like to do is begin with glucose, and we have to know that glucose enters a cell by something known as glucose transporter. So what I'm going to do right now is tell you guys how it starts, and it starts with glucose entering the cell by the glut, and then the glucose is in the cytosol. And for those of you guys who are not familiar with cytosol is cytoplasm. Now I want you guys to hear one thing right now before I go ahead and go further with this.
Molecules. Now, in order to explain exactly how glycolysis works, I'd need about an hour of your time in a giant cast of finger puppets, each playing a different enzyme. And though it would pay me to do it, I would have to use words like phosphoglucoisomerase.
Okay, let's stop right there. I know exactly what he's communicating about. He's talking about the glucose here and how it ends up being into a pyruvate form. And the way it goes about doing that is it has to go through enzymes. Now, real quickly, an enzyme is going to be a chemical substrate that deals with slowing down, speeding up the molecular reactions. So enzymes would be kinase, isomerase, hexokinase, phosphofructose. Those are considered enzymes. Now, you take glucose, and once it goes through the hexokinase enzyme, it becomes glucose 6P because it has taken what I consider phosphate from the ADP energy. Glucose 6P passes another enzyme that turns into fructose. Fructose is then converted into fructose 1,6P, and here we like to consider a triangle. Now, this triangle is going to show that fructose 1,6P goes into two different components. One component is going to be a dihydroxide ketone, and the other one's going to be 2-glyceride. The 2-glyceride is the one that we're worried about because that's the one that's going to be going into the biphosphoglyceride through the G3P dehydrogenase. Now, all of this is going to be transferred into what I consider more affiliated forms of glucose. All these are a form of glucose. Glucose, fructose is a form of sugar. Now from there, it's going to what's called enzymes that modifies it as time. Pretty soon, you're going to get into the 3-phosphoglyceride. Then you're going to hit a mutase enzyme that transfers 3-phosphoglyceride into 2-phosphoglyceride. In the analyze, an H2O comes out, then you have 2-phosphenylpyruvate, then it goes to a pyruvate kinase, which is an enzyme, and then from there you're going to have the final form, pyruvate. This is pyruvate, and this is pyruvate. What you see in common right now is the 3 carbons. That is called a pyruvate form 3 carbon. This right here is what you call the entrance to the mitochondria. Sometimes what they call the edge of the entrance of the mitochondria is kind of known as the membrane. So right here, the pyruvate dehydrogenase is going to actually take the pyruvate. It will extract a CO2 out of it, meaning that a carbon dioxide will come out of it, a CO2. And then from there, it's going to end up in this form where it adopts coenzyme A, and then it becomes coenzyme A with the SH at the end. Now, this right here is called the Krebs cycle, TCA cycle, and I'm trying to see if there's another name for it as well. Tricarboxylic acid cycle as well. These are the enzymes: aconitase, aconitase 2, isocitrate, Q-glutarate. I don't want to skip any enzymes here, but I really want you guys to know this one right here. And this one's going to be called the enzyme sonic dehydrogenase, which is going to be called the complex 2 enzyme. With this enzyme, I think it's really important you guys know, because this enzyme is attached to the electric transport chain as well, and also in the Krebs cycle. Big bonuses, big key parts to understand this. Here you have a stage of fumarate, and for the guys who aren't familiar, these are enzymes in between the boxes, and these are going to be called the stages of the carbohydrates. So with the stage of the carbohydrates, here's going to be 2 carbon, 2 carbon, 2 carbon. You see it loses a carbon here, and then it keeps on going through a continuous cycle all the way up to get malate, enzyme, and then oxalate. This is going to be the Krebs cycle. Now, once the Krebs cycle is further in detail, you're going to see here we got the electric transport chain with the enzyme 1, 2, 3, 4. So we got a lot of enzymes here, as we can tell. Complex 1 is an enzyme dealing with hydrogens. Complex 2 is attached to the Krebs cycle. So you guys can see how it plays a role in the electric transport chain and in the Krebs cycle. Coenzyme Q. You have a coenzyme SH above, and this can transfer into the complex 3 enzyme. And then here, complex 4. Bonus, complex 4 deals with the oxygen being converted into H2O. For those of you guys who understand information at Bloom taxonomy level, understanding oxygen, where it's going, where it's exiting, is going to help you out with the cellular equation. Now, since this is the animal cell, the equation you want is going to be the oxygen plus glucose, and then you want the outcome to be carbon dioxide and H2O. So here's glucose. That's going to be the source of energy that's going into the cell. And then basically what's going to happen from there, that a carbon dioxide is going to get released. Carbon dioxide is an outcome. Another income in the cell is going to be oxygen, and the outcome is going to be H2O. So this should help you out with the understanding of what molecules are going into the cell and what molecules are coming out of the cell. If one thing I would try to understand is try to understand the enzymes and try to understand the carbon dioxide. So we have what I like to consider 4 main components. We have the glycolysis, how the glucose is being converted from enzymes into a pyruvate. We have the pyruvate dehydrogenase, which tells you how it came to the acetyl coenzyme A. We have the Krebs cycle, TCA cycle, and we also have the electron transport chain. I honestly have not seen it in this full detail ever. This is going to be the greatest amount of detail I have ever released and seen. This actually contains every single thing known to mankind to me. Please contact me with information if you guys have any questions. I'll definitely put my email. I'm going to put my number out there as well. I already belong to 2 different biology club meetings. I'm going to post this video as well up there too. The reason why I want to make this video is because I put up my diagram onto a social network and I receive a lot of likes and a lot of feedback. Hopefully this video will help you explain the diagram and help you understand the diagram a lot more. And this is what I like to call the cellular respiration, cellular metabolism, without the protein, without the fatty acids, without the starch. Please feel free to give me any type of question, comments, and thank you very much for letting me play this video. Enjoyed it.